camera, please?
Okay. 1 minute.
I can see you.
Here we go.
Okay, good. Now I see you.
Here we go.
So I'm just
Okay,
going to say
great.
that Dr. Shu is a very talented scientist, specialized in virology, bioinformatics, and microbiology. And he works mainly in studies of plant-virus-vector interactions, virus immunity, population dynamics, and evolution of plant viruses. He is currently a PI at Nanjing University in China, coordinating a big team of scientists. Yi, thank you so much for coming today, helping us here in this course for the grad school. And please feel free to start whenever you're ready, okay?
Okay, yeah, thank you.
Thanks, Yi.
Hi, good morning, everyone. Thanks so much for Patricia's invitation. It's really cool to make this, available happen. So thank you. So today it's my pleasure to be able to speak with you and share my research findings from the past few years. So today I'm going to tell you a significant breakthrough in which we identified a novel geminivirus that caused soybean stick grain disease. This disease posed a considerable threat to soybean production in China, so leading to huge damage right now. So my research, as Patricia mentioned, so my research primarily focused on plant virology, as plant virus pose a significant risk to the safety and the productivity of agricultural crops. So in recent years, their impact has become more severe due to the climate change and modern agricultural practice. So this image shows that the emergence of plant virus disease in recent years, so highlighting the extensive damage they have caused to crops and vegetable production. So soybean is one of the most valuable legume crops globally, providing 25% of the global edible oil supply and also 2/3 of the global concentrated protein for livestock feeding. So with China being the birthplace of soybean, and I think it has a cultivation history of 5,000 years. So soybean, also known as shu in ancient time in China, so it's one of the most 5 important crops in the long history. So however, due to the increasing demand, China's soybean production is unable to meet the growing consumption need. So leading to over maybe, I think it's around 85% dependency on import. So as the world's leading soybean importer, China accounts for 1/3 of the global soybean usage. So I have to mention that the Brazil now is the main source of the imported soybeans in China, ranking number 1. So more than 50%, the soybeans that come from Brazil. So yeah, it's a huge amount. So in response to this demand, also China also the government has implemented various measures to encourage soybean cultivation, so resulting in a year-on-year increase in soybean planting area. However, alongside the expansion of the planting area, the issue of soybean disease and pest became also increasingly prominent. So soybean virus pose a major threat in yield production. Currently, more than 100 viruses are known to infect soybean, and of these, at least 47 has been detected in naturally occurring infections in field. So in recent years, the Huanghe region of China, it's in the middle of China, so has witnessed the emergence of soybean stick grain syndrome, so SGS. So leading to a significant soybean yield losses. In some area, the seed yield has been completely wiped up. So we performed field experiment showing that when the incidence rate exceeds 50%, the soybean yield is reduced by 2/3. So it's a big problem. It's a big problem right now. So soybean stick grain seedling is characterized by the delayed senescence at harvest, resulting in the retention of green leaves and whole plant, poor pod production, and started seed development as seen in the image. So it just keep growing when during the harvest season. So also the seeds are severely affected. So that's really bad to the production and the yield. However, the exact cause of disease remains unknown. Many hypotheses have been proposed, including pathogen disease, pest, stress, or environmental factors. So, however, the lack of the clarity regarding the pathogen responsible for this disease has hindered the development of effective prevention and control methods. So my objective of this study was focused on identifying the etiology of soybean stick grain syndrome. We successfully detected a novel geminivirus species in soybean samples, showing the stick grain symptom. So through fulfilling the Koch postulate, we confirmed that the infection caused by a novel geminivirus. So this geminivirus is responsible for the soybean stick grain syndrome. Additionally, we determined the insect vectors of this novel geminivirus. So that's also we conducted the epidemiological evaluation based on the 4 years of field investigation in multiple locations of China. So initially, our primary objective was to determine the percent of plant virus in soybean sample displaying the stick grain symptom. In 2019, I just returned from Cornell. So I have no project or something. I just collect many samples from the field. So we collected numerous stick grain soybean samples from various locations in Huanghe region of China. So this plant showed symptoms such like leaf crinkling, staining, flat pod, and poorly developed seed. So furthermore, we observed a high population of insects with piercing, sucking mouthpart in the field. All these observations strongly suggest an association between soybean stick grain syndrome and virus disease. So those samples were mixed and identified the virus with small RNA-based sequencing. So as Patricia mentioned, that I had been working on this technology for more than maybe 15 years. So we have a lot of experience. So by using this technology, so after using this high throughput sequencing, so we identify around 66,000 cantiques assembled by the Webword and other software. So one cantique with a length of 300 nucleotides, showing the high sequence similarity to a bigomovirus C1. So it's a bigomovirus. So then, to obtain the complete viral genomic sequence, a pair of back-to-back primers was designed, and the virus genome was subsequently cloned and for sequencing. And the complete viral genome sequence was determined to be 2762 nucleotide in length. It shows typical genome structure of geminivirus. So including 2 offs, V1 and V2 from the variant strand, and also 4 offs from the complementary sense. The size of the intergenetic region was it's around 2 183 nucleotide, which is similar to that of the most virus. So within the intergenetic region, a nanonucleotide showing in the red color here. It was also found, and the intergeneric region of this geminivirus contains a TATA box, which, based on the soybean symptoms observed in the field, the virus was tentatively named soybean stick green associated virus. So I want to take some time to introduce the technology because Patricia mentioned that showing some technology or the method we used during the plant virology research. So this technology is based on the post-transcriptional gene silencing. So PTGS played evolutionary conserved role in gene regulation and defense against the invasive virus. So the enzyme, including the diethyl agonate, catalyze the digestion of virus RNA and generation the small RNA that mediated antivirus immunity during the infection. The accumulation of virus-derived small RNA is a common feature of an active immune response to virus infection. In diverse, many plant or insect host species, and the virus small RNA are overlapping in sequence, which can, this is very important, which can assemble into long context, of the invading viral genome by the next generation sequencing platform. So based on this, we can identify the virus. So actually, this method was first established by Shoei Lab from UC Riverside in 2009. So they identified the virus from the insect. So we followed up and also established a very similar platform to identify virus from plant. So noteworthily, this method can both identify the known virus and novel virus. So also I have to mention that compared with RNA-seq, which is pretty similar to all the audience. So only virus that can replicate in the host can be detected by small RNA sequencing, because only replicating virus can generate the double-strand RNA stage, and also can produce small RNA. If the virus can't replicate, so no small RNA could be found. So this is the basic for this technology. So here I show you a flowchart representing different approach for the analysis of the high throughput sequencing data for the detection of plant virus. So it's a pretty strong tool for identify pathogens right now. No matter in plant biology or animal or even in the COVID-19 or something like that. The high throughput sequencing technology is very powerful now. So then I return to my research. We identify the soybean stick green associated virus in the samples with the stick green symptoms. So we also get the genome, and also then we do the analysis, right? So the phylogenetic analysis revealed that soybean stick green geminivirus SOSGV formed a distinct cluster separated from any existing genus in the family of Geminivirus. So it clustered mostly close to the meldovirus in the meldovirus genus, but in unclassified groups. Next, we also analysis the segmented sequence suggests that SOSGV might be a recombinant virus. which is genetic ancestry possibly derived from recombination event, including different genes in the geminivirus. So specifically, genomic fragment from mother virus and begomovirus linkage, and they recombinate together and get the novel SOSGV. So notably, the right portion of this virus from maybe including the V1 and V2. Yeah. Yeah, the right part of this virus, they had very little sequence similarity with any sequence in the NCBI nucleotide database. So consequently, the origin of this novel virus remains unclear. So its parental virus has yet to be identified. It's really a pretty novel virus. So furthermore, we demonstrated that all collected. Yeah, we using the same plot analysis, also showing a similar story that the virus is properly formed by intergenetic recombination. So furthermore, we demonstrated that all collected 27 samples testing positive for SOSGV showed 3 green symptoms, including delayed leaf senescence, flat pod, and abnormal seed. So it's very consistent with all the samples. So this finding strongly supports the high association between SOSGV with the soybean stick green syndrome in the Huanghe region of China. To determine whether this novel virus is a causal agent of soybean stick green disease, we constructed an infection clone. And additionally, we developed an efficient inoculation method to introduce SOSGV to soybean. So our result demonstrated that over 90% inoculation efficiency of SOSGV in various soybean cultivars, so such as Williams and also some native cultivars in China. So to confirm the systemic infection and the efficient replication of SOSGV in the inoculated soybean plant. So we also performed the thousand we also conducted 1,000 blot hybridization experiment. This result provide evidence of successful infection and robust replication of SOSGV within the inoculated soybean plant. So interestingly, 4 months after inoculating SOSGV, all soybean plant with mock treatment become yellow and get mature, and with mature pod and seed. In contrast, all SOSGV inoculated plant tested positive for SOSGV continued to display the 3 green symptoms, including delayed leaf senescence, an increasing number of abnormal seeds, and numerous flat pod. The symptoms observed in plant inoculated with SOSGV infection clone was indistinguishable from those found in the field. So furthermore, the exact effect of SOSGV on seed yield per plant was also counted. So the result showed that infections by using cloned SOSGV had little effect on pod number, but they significantly increased flat pod number, so reduced total seed number, and decreased weight per 100 pod and also seed size. So this effect was consistent with the characteristics of soybean stick green syndrome observed in the field. So subsequently, we tested more than 100 soybean cultivars, and all of them exhibit stick green symptoms after SOSGV infection. So this experiment so strongly supports that SOSGV is the causal agent of soybean stick green syndrome. Moreover, we found that SOS-GV could also cause stick green-like symptoms on Nicotiana benzimiana after inoculation of SOS-GV infection clone. So these symptoms also included delayed leaf senescence and a higher population of empty pod compared to plant inoculated with the empty vector control. So collectively, this result provides further support for the ability of SOS-GV to cause soybean stick green syndrome. So next, we explored the factors contributing the epidemic spread of this disease in the field. So initially, we determined the natural host range of the SOS-GV using our efficient inoculation method. So we demonstrated that SOS-GV can infect legume crops such as peas, chicken peas, and also Chinese milk welch, as well as the plant from the Solanaceae plant family, including tobacco and some other plant. So overall, we confirmed that SOS-GV has the ability to infect more than 20 plants, and also wheat, which belong to several families of plants. So the transmission mode of the SOS-GV was also investigated. So interestingly, we found soybean seeds from field sampled with the stick green symptoms carried SOS-GV, indicating the potential transmission of SOS-GV through seed. So to employ this possibility, we collected more than over 100 seeds. This plant from the SOS-GV infected plant. So we performed the grow out test. However, PCR analysis of the resulting seedlings showing that none of them tested positive for SOS-GV. Subsequently, we deserted the seed coat from the infected plant. So just the seed coat, and found that SOS-GV was present in 100% of the seed coat, but not in the endosperm and also the embryo. This explains why SOS-GV can't be transmitted through seed. So as mentioned earlier, the outbreak of soybean stick green coincide with a high population of insects with piercing sucking mouthpart in the field. So by collecting these insects, we identify whitefly as the dominant insect species in the Huanghai region of China. So, the statistical analysis revealed that over 98% of these whitefly belong to the Q biotype, so indicating it's prevalent in this region. So, despite this, our repeated experiments demonstrated that whitefly, both Q and B biotypes, and also bean bugs and some other insects failed to transmit SOS-GV. So we repeat many times. So to identify the specific vectors responsible for SOS-GV transmission, we conducted laboratory tests using various insect species, which we collected from the soybean field in the Huanghai region of China. Finally, we identified the leafhopper species Oryzurus orientalis as the vectors of the soybean SOS-GV. So this insect also named the common brown leafhopper. It's not native China. So it's firstly found in Australia, the South Korea, and also Philippines. So also the Malaysia and from the Pacific Island, I think. So molecular characterization through cloning and sequencing the CO1 gene further validated the identification of these leafhoppers. So as shown in this image, after 3 months of transmission by the common brown leafhopper. The infected plant also displayed symptoms characteristic of the stick grain, including checker and dark green foliage, an absence of flat pod and seed with abnormal feed filling. So it's important to note that also I mentioned earlier, it's not native to China. Yeah. So further research is needed to investigate the distribution, host range, and the relationship between the percent of the common brown leafhopper and the incidence of soybean stick grain syndrome in China. So in recent years, in collaboration with numerous scientists from the China soybean research system, we have collected more than 500 samples exhibiting delayed senescence during the harvest season. So these samples was obtained from 17 distinct geographic regions across 8 provinces in China, revealing the expanding range of SOS-GA. Also, the phylogenetic analysis revealed that a significant genomic variation between the isolate from Luliang and Shijiazhuang regions compared to those from other regions, placing them in a distinct clique. Furthermore, our epidemiological assessment of the SOS-GA indicate ongoing genetic differentiation. So to gain deeper insight into the origin, evolution, and epidemiological dynamics of SOS-GA, we established the time of the most recent common ancestor, TMRCA, and the nucleotide substitution rate of the complete genomic sequence using the BEAST software. So yeah, as shown in the right, so we get the exact the mutant rate and also the TMRCA also SGA was also clarified. Furthermore, so the mechanistic investigation also SGA induced soybean stick grain symptoms were also studied. So we showed evidence that SOS-GA infection disrupt the partition of sugars between cells and the sink tissues, leading to the accumulation of the sugars in the south leaves. Yeah, most of the sugars in the south leaves so are retained in the south leaves, and subsequently causing the delayed senescence. And the precise mechanism by which SOS-GA impaired the translocation of sugars from south to sink is currently under investigation. We aim to uncover this exact mechanism and look forward to sharing our findings in the near future. So yeah, it's clearly showing that the SOS-GA can disrupt the sugar transport. So most of the sugar are returned, just returned in the leaves, and they can't translocate to the sink tissues like seed and also the root. So that's why you can see the plant showing the stick grain because they have a lot of sugar and nutrient and something like that. So we will discover, we will uncover the exact mechanism about how SOS-GA disturbed the sugar translocation. So I would like to express my gratitude to my 3 dedicated graduates, Ruixiang, Rong, and also Ruoxin for their hard work in this study. So I'm thankful to Xiaorong for providing valuable ideas for this research. So as well our collaborators from Yuanchao Xueping's lab. Yeah. So moreover, I extended my appreciation to the funding sources that supported this research. So with that, I would like to conclude and also address any questions you may have. Okay, thank you.
Hi, very nice talk. Thank you so much.
Thank
You know, you guys in China work so hard and so fast that it makes it very difficult for us to compete with you guys, right? you just did everything, right? I saw there's some unpublished data, but you guys did already everything. It's very interesting how you guys work. So,
thank you. Yeah, in China we have a new word for this situation. It's 卷. It's just very competitive for everyone.
yeah. And
Pressure,
how many
yeah,
people you have in
yeah.
your lab today?
12 people.
12. Wow, it's a lot of
Yeah.
people.
five PhD candidates and also
Cool.
yeah.
Very
It's a
nice.
small group here.
Yeah, it's small. Oh my god. Okay.
Yeah.
It's a lot of people for me. You know, if I had 12 in my lab, I think I would be crazy right now.
Yeah.
So, let me just go back to when you explain how you guys figure out that this is a novel virus, right? So you did the high throughput sequencing, and then you did a lot of bioinformatics there, and then you found out that this is a novel and not a mutation of something that is already out there. So can you go back to that again and just let us know how this magic happens?
So, yeah, of course. So yeah, can you see my screen?
Yes.
Okay.
So, okay. But I can see. I can see the slides. Yeah, we can see.
Okay.
Now. Right.
Okay. Yeah. So actually, initially we just collected samples from the field, right? So yeah, this sample, it seems like virus infection. It's some leaf crinkling draft also for that pot, but never. Some people also detect the known virus. The maybe 3 or 4 virus that's known to infect soybeans, but they failed to detect any virus in the sample. So they said, oh no, the soybean green syndrome is not caused by the virus. So yeah, as a plant biologist, yeah, we are very excited to see the symptoms like this. So I just collected samples from the field. So yeah, because my deep sequencing is really my area that's very familiar with this area, so this technology. So we just isolate the total RNA from the samples with symptoms, right? So we combine these samples, the total RNA, and then we separate the SMID from the total RNA, and then SMID was constructed the library, which I show you in this figure. Maybe it's not so clear. So
actually, I was thinking about that slide when you show. I think not this right one. Bottom. I guess that other one where you show the proteins and the map of the virus. I think it's one more. Yes, this one. I guess here you said something about how you recognize the genes and proteins, right?
Right. So this is the software named Simplot. So yeah. So for example, the red color represents the SOS-TV, and the other two represent two representing virus in different genus of geminivirus. Gemini virus. It's from different genus. So you can see, yeah, from here, yeah, there are no sequence similarity. Yeah. So but it should more close to, yeah, for the blue one, and also so for the right for the right part of this the genome sequence showing high similarity with the green. It's a typical Begonia virus. Gemini virus has 14 genus. So yeah, 14 genus. So I just showed that. So this virus is just a recombinant virus that comes from two genus in the Gemini viridae. So it's from
Which ones again? It's begomovirus?
Begomovirus.
And the other genus?
The other from the meldovirus. It's also a genus in the Geminivirus family. So,
okay.
Yeah.
Cool.
Yeah.
So it's a recombinant from these two.
Right.
Cool.
Yeah. It's just a recombination. Actually it's a very long fragment recombination. It's not just small recombination. It's a very long it may be half, maybe more than 1,500 base pair BP. So it's a pretty long recombination event.
Okay.
So
yeah, we also have some questions here in the chat. So Simone is asking, first she acknowledged you, right? Thank you, Dr. Shu, for the interesting talk.
Mm-hmm.
And is there any other virus infecting the stay green plants? For 20% of the surveyed plants, you said that SOSGV was not present.
Right, that's a really good question. So, yeah, I will show you a slide to explain this. Here we go. So I first tell you the conclusion that over 80% of soybean stick green samples contain SOSGV. So there's still 20%, right? So they also showing stick green symptoms but no SOSGV. So that's very interesting, and we found that any factors that can disrupt the south zinc, the sugar translocation will cause stick green, no matter insect feeding or the heat stress or other environmental stress. So actually, we didn't find any virus in the soybean, any other virus besides except SOSGV in soybean stick green samples. But we do find there are some samples with stick green symptoms, which they didn't. There's no SOSGV. So that's my answer.
So maybe stay green is not always caused by virus, right? The symptoms.
Yeah, that's very important. So we do the epidemiological, the evaluation, the assessment. We showed that 80% of the soybean stick green syndrome was caused by SOSGV. It's caused by the virus. But any other factors that can affect the salt zinc sugar translocation will also cause a similar symptom. But this is not a big mess. So only the virus, that's really a big big problem here. So I agree with you, because anything that can disrupt the zinc salt translocation, the sugars, the translocation changed, the plant will show stick green symptoms, I think. Yeah. Sure, but we didn't find any virus in the soybean stick, other virus in the soybean stick green system. Yeah.
Interesting. So, Murad's asking, did you do more work on the virus vector interactions? Is it a persistent virus, maybe replicating the vector? He wants all the answers, right? Did you test other vectors? Maybe. I guess he's trying to say that he wants to collaborate with you.
Yeah, that's really cool. Yeah, we just we just report the insect vector, but the transmission mode of this this SOSGV with the leaf hopper we didn't know. We non-persistent or the replicating or something. We have no idea about that. We are working on this right now. So
I'm pretty sure you guys are. I know you are.
Yeah.
So Murad, you want to say something? The microphone, or is okay?
Yeah, it's interesting.
Yeah. Hi.
Thank you, E, for the talk. It's very interesting, and it's always interesting it reminds me our story with the virus that we detected here in Israel, the virus in pepper, if you remember.
Yeah.
We detected a virus that was also a result of recombination between. It's a polarivirus.
Right.
And it's a result of
Right.
And it's a result of recombination between two other viruses. One
That's really cool.
One parent is present in Israel, and the other parent we don't know where it came from.
Wow. It's the same situation. For me right now. So right.
But the interesting thing about this virus that it belongs to polariviruses, which are transmitted by aphids, and this new virus is transmitted only by whiteflies, not aphids.
Right. That's really cool.
And it's also transmitted by only one biotype, with the B biotype, not the Q. So so it's a kind of a similar, but it's not
actually.
In your case, it's not a whitefly.
It's not a whitefly. So we tried many times, but it failed to transmit. I have to mention that whitefly is very, the population is very high right now in China in the soybean field. If the virus can be transmitted by whitefly, it's a big disaster, I think. So luckily, so it can't. It can't.
Are there whitefly transmitted viruses in soybean? I think in Brazil there is one, right?
But in China,
we try CPMMV, Murad, is transmitted by the whitefly to soybeans as well, but they are not important in the soybean here.
But there are others begomoviruses that are transmitted by whiteflies here in Brazil.
Okay.
They're not very important, but they were identified.
Soybean is more like a reservatory, right? Reservoir here. So the viruses are important to common beans, not to soybeans, but they go here and there, right?
It's really cool. So we didn't find CPMMA is also sometimes occurred in China, but last year we didn't find any CPMMA, and also I know it's transmitted by B biotype, right?
Okay.
Whitefly. So we didn't find that. So luckily.
Thank you. It's interesting.
I will talk with you later about Murad. So actually, one of my colleagues in Zhejiang University, they also found a similar story like as you present in the GAV paper. So maybe I can talk with you later. So
okay.
It's from a different host. So I just recommend them to just tell them to read your paper. So it's a really classic paper.
Okay,
so I keep asking my students here if they would like to go to China or to Israel. Do you have room there for them?
Help you? So, actually, I have. If anyone wants to come, I have. We just got a grant, so if anyone wants to work on whitefly transmitted viruses,
that would be very nice. I say that as a like I was kidding, but actually it's really interesting because there's a lot of things that we can share and experience that we can network and everything. And it's a very nice cultural experience as well, right? To spend some months in a different country, in a different culture. And you guys are also welcome to send students here. We will be happy to receive them as well if they want, okay? I don't know what they would like to do here because, things there seem to work very faster than here. So, but maybe, right? We don't know. Maybe they want to come sometime.
Here. It's good for them. Good for them,
Yeah. So any more questions, guys? Students are very quiet here. I don't know if they are shy or if they really don't have questions. But okay, it was very nice. And thank you so much for that. I think I'm gonna finish the recording here, and then we can talk about the other things that we want to talk very quick. Okay, about working.
Okay, thank you.
Thank you, everyone.
Okay.
Thank
So I'm
you.
gonna,
Thank you. My pleasure.
yes,
Bye-bye.
it was really nice.